Okay, could somebody confirm you can hear me?
I can hear you.
Thank you, Haley. Should be able to see my screen now. Excuse me. Today is Tuesday, May 28th. And we are supposed to be covering chapter 14. I don't think we're there yet. I think we need to finish chapter 8. Let me warn you, there's going to be a lab this Wednesday. Quiz 5. Quiz 5 will cover chapter 8 and lab 9. So quiz 5 will cover the least amount of material of any of the quizzes. Let me scroll this over. There is no lab for today. You should be working on your unknown project. Any questions about what we're going to do?
Yeah, at the end of the lecture, is it okay if I ask you regarding my lab, my unknown project, I mean?
Um, I would rather you asked it during the lab. I will be logging in for 15 minutes during the lab.
Okay, sounds
They
good.
only
Thank
have
you.
10 minutes in between, so.
Okay, sounds good.
Okay. All right, great. I have that we had covered transduction. Is that where we left off?
Yes, I believe so.
Okay. So to remind you, there are three ways where DNA can move from one cell into another cell, and that's transformation. I got a picture of it here, where DNA moves out of one cell and is in the environment and then gets taken up by a recipient cell. The second way is by conjugation, which can happen in two ways in gram-negative cells. There's actually a cell which has a sex pilus. It has a plasmid which makes the sex pilus. The sex pilus then goes out and skewers an F minus cell and then allows the plasmid to replicate in this cell, and it sends the plasmid over the sex pilus into this cell. That will then convert this cell from an F minus cell to an F plus cell, because this cell will have the plasmid as well as will grow the sex pilus. The other way that conjugation can happen is with gram-positive cells, and that's a mating bridge where two cells come up together and the cell wall disappears in one spot. And then the cytoplasm can actually fuse, or I should say the cell membrane actually fuses. So the cytoplasm of this cell is now connected to the cytoplasm of that cell. And DNA can move from one cell to another. Usually it's plasma DNA that will move. Any questions about that? And then the third way that cells can pick up new DNA is by transduction, where a virus moves DNA from one cell into another cell. Let me blow this slide up. So the virus replicates in this cell, destroys the chromosome, and then replicates its DNA. But the virus can make a mistake, and instead of incorporating the viral DNA, it incorporates the DNA of this cell. And then this virus will inject the DNA of this cell into that cell, and that will give this cell new DNA. All right, we've talked a little bit about plasmids, so let me talk a little bit further. They are simply a small circular extrachromosomal DNA, meaning a circular piece of DNA outside the chromosome. They only contain a few genes, like this one right here. Let me blow this up. Has 1, 2, 3, 4, 5, maybe 6, 7 genes on it. And a chromosome, as you know, has thousands or even tens of thousands, or maybe even if you're talking about a human cell, hundreds of thousands of genes on it. I think it's mostly tens of thousands of genes on it. Whereas the plasmid is a real small piece of DNA, which only has a few genes on it. The plasmid can replicate independently of the cell's chromosome. And I've actually already showed you that. Let's go back. When we were talking about conjugation, the F plasmid replicated in this cell and then sent one plasmid into the neighboring cell. That replication of the plasmid was independent of replication of the chromosome in this cell. Where are we? Plasmids don't tend to be in human cells, and actually most eukaryotes don't have plasmids. But yeast cells, which are a eukaryote, do have plasmids, and then plasmids can be found in all prokaryotes. There are different categories of plasmids. You don't need to know the categories. You just need to know that the plasmids can be of different types, different categories. Let's move on, talk about a transposon and insertion sequences. Transposons are a region of DNA which actually can move from one region to another region of DNA. And they're called jumping genes because literally they jump around in the cell. For humans or a corn, which I'm more familiar with corn, transposons were actually discovered in corn, by the way. Transposons can move around from one chromosome to another. A transposon is a segment of DNA which has always the transposase gene in it, and the transposase gene cuts out this region of the DNA and then can insert this region of the DNA in a new place. And obviously the transposase gene has to cut the DNA and then ligate it together to put in the transposon. I think the transposase gene also will reanneal the DNA. So this DNA piece right here and this DNA piece right here, the DNA will then be connected together from this part of the DNA to that part of the DNA. A transposon always contains the transposase gene and then the inverted repeat, which are nucleotides which are actually inverted. ATC, ATC. It's thought the inverted repeats help the transposon insert itself into a new spot in the chromosome or in the DNA. That's a simple transposon. All it has is the transposase and the inverted repeats. But there can be complex transposons which carry other genes, like TN5 is a complex transposon, and it can move kanamycin resistance gene around in the DNA. A complex transposon usually has a transposon over here on this side of the inserted gene, and then another transposon on this side of the inserted gene. And then this unit acts as not as two transposons, it acts as one unit and acts as a one transposon. This is important because you can have the kanamycin resistance gene moving around in the cell. And if the kanamycin resistance gene is in the chromosome, it can then move into a plasmid And plasmids are more likely to be shared between cells than the chromosomal DNA. So the kanamycin resistance gene will then be more likely to be shared among other cells in the population. Any question about any of that? Here's a transposon, which actually moved from the chromosome to a plasmid. All right, any question about transposons? They are important because obviously they act as recombination. I think I'm going to talk about, I've already talked about recombination, and that is that recombination shuffles up the genes and makes new genetic combinations where gene A and B, which were not together, they can now come together and then be passed together. And transposons can shuffle up the DNA even more so than recombination. And then transposons can also speed up the transfer of a gene from one cell to another by the transposon moving the DNA from the chromosome into a plasmid. And plasmids are just more likely to be shared between cells than chromosomal DNA. Any question about any of that? All right, so let's end this lesson talking a little bit about genes and evolution. Genetic variation is the difference in the genes. Genetic variation can only, new genetic variation can only come about from mutation. If the gene does not exist in the population or in a species, it cannot come into the population or species until it evolves by mutation. Crossing over as well as transposition shuffles up the DNA, creating new genetic combinations, not new genetic variation, but just bringing different genes together. And then transformation, conjugation, and transduction can allow cells to share DNA as long as the DNA pre-exists. As long as the DNA is in the species, transformation, conjugation, and transduction can move the DNA from one cell into another cell. This can drive evolution of the species by natural selection. Any question about any of that? All right, if not, that's it for chapter 8. While I'm shutting down chapter 8, let me go ahead and make that available to you. Let me make sure that previous one was available. Yep, it's available. All right, so let's move on to, to chapter 14, the principles of disease. As is always, I give you an introductory slide, which shows you the major goals and a rough outline. First, know the terms, pathology, etiology, and microbial antagonism. I've only got 3 terms here, but on chapter 14, if you learn the objectives and learn the, what am I trying to say, the terms of chapter 14, you'll actually learn about 50% of the material, because chapter 14 is really heavy on introducing terms. And then terms are very important to this chapter. They're always important to the chapter, but in this chapter, they're really important. So learn the terms for chapter 14. 2, understand the relationship between the normal microbiota and its host. And 3, understand the stages of disease development. Disease normally goes through a specific stage of development. And then 4, understand what nosocomial infections are and how to control them. Any questions about the goals and the rough outline? All right. So pathology is the study of disease. Etiology is the study of the cause of a disease. For example, the etiological agent of the disease anthrax is the bacteria Bacillus anthracis. Pathogenesis is the development of a disease. Infection is the colonization of the body by pathogens or the presence of microbes where they are not normally found. Like E. coli is normally found in our gut, and it normally doesn't cause any problems. In fact, it's beneficial to us because it helps us digest food. However, if you find it growing in somebody's ear, like a child's ear, or you find it growing in the urethra, it can cause an infection where the microbe is not normally found. Disease is an abnormal state in which the body is not functioning normally. When we're talking about disease, you should realize that only a few microorganisms are pathogenic. Only a few microbes can cause disease. Most microbes do not cause disease. Some microbes are even beneficial, such as the microbes of the normal microbiota. Not all of them are beneficial, but many of them are. Like I mentioned, E. coli helps us to digest our food. The normal microbiota is a very large community of microbes that are found in association with animals and plants. These communities are known as the normal microbiota, microbes that permanently colonize a host and then permanently, or at least tend to permanently live with that host. For example, when I was first born, Staphylococcus epidermidis colonized my skin, and it has stayed on my skin and has continuously stayed on my skin and will be on my skin until I die. Obviously, when the skin flesh rots away, the Staphylococcus epidermidis will no longer be there, but until that time, it'll be there. And it was continuously been on my skin. The human normal microbiota is called the human normal microbiota, and that will differ at least somewhat from the normal microbiota of other animals, like a cat normal microbiota. Like cats have, let me see if I can, Bartonella henselae as part of their normal microbiota. Bartonella henselae is not a part of the human normal microbiota, and if somebody gets Bartonella henselae, it will cause disease called cat scratch fever. Any question about any of that? However, cats and humans do share a lot of microbes, like Staphylococcus epidermidis is on both species. The normal microbiota is only found in certain locations, and then disruption of the normal microbiota host balance appears to be associated with disease and impaired immune function. An example of disruption of the normal microbiota host balance is when you take antibiotics. Obviously, you're taking it to stop disease, but when you take the the antibiotics, you disrupt the normal microbiota, especially of your gut, but essentially everywhere in your body. And a lot of those normal microbiota die. Oftentimes, when people take antibiotics, they will either have diarrhea or they will become constipated. And that's because the normal microbiota host balance has been disrupted, and it'll have to reestablish itself. And usually it reestablishes itself pretty quickly, and then the diarrhea and the constipation usually quickly go away. Obviously, that doesn't always happen, but usually it does. And when you take antibiotics, you don't always get diarrhea or constipation, but sometimes you do. Any question about any of that? The normal microbiota is only found in certain places in the body. Let me blow this up. Such as the skin, the the mucosa that's open to the outside, such as the eyes, the mouth, the throat. The gastrointestinal tract has a lot of normal microbiota. Yeah. The lower urinary reproductive systems, meaning the lower urethra on both males and females, have normal microbiota. Usually, deep in the respiratory system, like the lungs, usually do not have normal microbiota, as well as, at least in males, the bladder and higher up on the urinary reproductive system does not have a normal microbiota. And I should say it's never been studied, males in the bladder, but certainly the kidneys and the ureter do not normally have normal microbiota. Obviously, the urethra does. Places where microbes are not found include internal to the body, such as the blood, the cerebrospinal fluid, the muscles, the nervous tissue, and the bone. The normal microbiota differs from transient microbiota in that the normal microbiota, once it colonizes the skin, it permanently takes up residence on the host, essentially for the lifetime of the host, like Staphylococcus epidermidis on my skin. Transient microbiota may be present for days, weeks, or months, but unlike the normal microbiota, transient microbiota do not take up permanent residence. For example, a transient microbiota that I once had was I went and visited my aunt, and she insisted on hugging me, even though she had a Staphylococcus aureus infection on her skin. And she probably thought that she was fine because she was taking antibiotics for Staphylococcus aureus. But I actually was testing myself at that time every term to look and see if I happened to have Staphylococcus aureus growing on my skin. And I never had it on my skin until after my aunt hugged me. And I looked at that and said, geez, I must have picked up Staphylococcus aureus when my aunt hugged me. Fortunately, the next term and future terms when I sampled my skin, Staphylococcus aureus was no longer there. It had gone away. Probably what happened was the normal microbiota took up space and forced the Staphylococcus aureus to go away. meaning it didn't have any more room or space or food, and the Staphylococcus aureus died out and then did not continue being present on my skin. So that was a case of transient microbiota. And I was quite pleased that it was only transient, because if you do not know, Staphylococcus aureus is a frank human pathogen. People who have Staphylococcus aureus on their skin typically have skin boils every once in a while and other skin lesions, namely skin boils. So if you're a person who gets boils on your skin every once in a while, you probably have Staphylococcus aureus as a part of your normal skin biota. Only about 20% of Americans have Staphylococcus aureus growing on their skin. 80% of us do not. However, Staphylococcus aureus is more likely to be present in the nasal cavity. I think 30 to 50% of Americans have Staphylococcus aureus growing in the nasal cavity. And fortunately, in the nasal cavity, for whatever reason, it tends not to cause harm. On the skin, it tends to cause harm, not all the time, but some of the time. Any question about any of that? All right, the normal microbiota on the host. Humans have 3 times 10 to the 13 human body cells. And growing in and on us, we have 4 times 10 to the 13th bacterial cells. This means there are more bacteria cells in and on us than there are human cells. And that begs to ask the question, are we more micro than we are human? And don't let that freak you out too much. Any question about any of that? So our normal microbiota, there are more cells of the normal microbiota than there are human cells. Here are some pictures of the normal microbiota. The orange cocci are cells of the nasal epithelium. These are not rods. Those are actually, oh, what do you call that? Villi, is that what they're called? That's part of the nasal epithelium. That's our part of our normal cell, and they move to move the mucus of the nasal epithelium. Here is some bacteria on the stomach. A Helicobacter pylori. I don't know if that's Helicobacter pylori. It might be, but that's some bacteria in the stomach. And these right there, those are not, this is part of our stomach. These are villi in the stomach. And that's obviously the, I don't know if you call it the mucosa or that's the stomach lining. Here's some normal microbiota in the intestine. This right there is not normal microbiota. That's actually food that's being digested. And right there, that looks like a rod. This looks like a coccobacillus. And that cell kind of looks like a cocci. It's not. It's probably a coccobacillus, just of shorter shape. But those look like cocci. Part of the normal microbiota of the intestine. Like I said, there's tons of normal microbiota in the intestines. I think I mentioned that the wet weight of your feces, about 50% of it is from normal microbiota. All right, we found some human normal microbiota in some surprising places fairly recently. Like they've discovered that there is normal microbiota in the female bladder. I've looked to see if there is normal microbiota in the male bladder, and nobody studies it in males. And as far as I know, the answer is nobody knows because nobody studied the male bladder. It is possible that the female bladder has normal microbiota and the male doesn't because the urethra of the female is much shorter. But the point is the bladder, at least in females, is not sterile. There is some normal microbiota in the bladder. And this also was a surprise, and that is that breast tissue in mother's milk is not sterile. There is normal microbiota in the mother's milk even before it leaves the mother. And the breast tissue also has normal microbiota inside it. Any question about any of that? That actually is fairly important for a baby because when the baby first, I guess, starts nursing, obviously he will get the normal microbiota from the outside of the nipple, but he's also getting the normal microbiota that the mother puts in the mother's milk. And that helps the baby pick up its normal microbiota for the gastrointestinal system, as well as help the baby in digesting food. So the normal microbiota in mother's milk is actually important for the baby to make sure that the gastrointestinal tract is colonized with the correct microbes. And if you don't know, a colicky baby, meaning a baby that tends to cry a lot, they've discovered that most colicky babies have C. diff growing in their intestine. What happens is C. diff, which has endospores, so once the baby gets it, it's very hard to remove, is something that you don't want to get in the gastrointestinal tract of the baby. I'm not sure where the baby got it, but C. diff is fairly common, and so are the endospores of C. diff, and you only need a few. And if the baby picks it up originally, then the baby is going to be having the C. diff growing in the gastrointestinal tract, causing the baby discomfort. I think it causes gas and bloating, but I'm not certain what it does, but it can also cause an infection. Anyways, that causes the baby discomfort, and that's why the baby cries a lot. Usually with C. diff, the C. diff will go away on its own, but this could take weeks or months. And I'm not sure if doctors now will prescribe antibiotics to remove the C. diff. If you don't know, you need to prescribe antibiotics twice to get rid of C. diff, and that's because C. diff creates endospores, and the endospores will not be killed by the antibiotics. One thing you should never give babies is honey, because honey has endospores in it. Any question about any of that? Including it has a low numbers of a toxic endospore, which because the baby is so small, even low numbers could be toxic to the baby. Whereas honey, the toxic endospores will not harm an adult because they're much larger, meaning the toxic endospores are really small numbers in honey. And so for adults, it's safe to eat, but you shouldn't give honey to a baby. Here's a place where they said that the finding the normal microbiota was surprising, and I wonder why it was that they found that the placenta has a normal microbiota. And why I say that's not a surprise to me is the female reproductive system is open to the outside. And so that the vagina and up the vagina can get normal microbiota. And so the placenta shouldn't have been a surprise if people would have thought about it, because it's obviously connected to the female reproductive system. But the placenta has a unique normal microbiota, which only occurs when the female is pregnant, the placenta. And when the female is pregnant, the placenta has a unique microbiome, which is found nowhere else in the human body. Any question about any of that? All right. The normal microbiota does establish a relationship with the host. Generally, this relationship is beneficial. The normal microbiota can help the body, and one way it helps the body is by microbial antagonism. Microbial antagonism is competition between different species of microbes. So the normal microbiota can prevent the growth of a pathogen by occupying the niche that the pathogen might occupy. For example, I've got Staphylococcus epidermidis on my skin. So if Staphylococcus aureus were to happen to fall on my skin, it might fall on part of the skin where Staphylococcus epidermidis already is. And then the pathogens, Staphylococcus aureus, cannot get established because the niche is already occupied. It can also compete for resources. Staphylococcus epidermidis can use up all the resources around the area where Staphylococcus aureus falls. Staphylococcus aureus now has no resources, and so it will not grow and get established on the skin. The normal microbiota can also produce acids, which can make the environment less hospitable for a pathogen to get established, like the normal microbiota of our urethra, of our mouth, and of our skin make those environments more acidic. That makes the environment less hospitable to a human pathogen. And if it's not hospitable, then the pathogen cannot grow and get established. An example is that the skin is around pH 5 to 5.5. Some pathogens, not Staphylococcus aureus, but some other pathogens, if they were to fall on the skin, they would find the skin to be too acidic to grow. If you remember, most things like growing between pH 6.5 and 7.5, and the skin is below that pH. Some normal microbiota can also produce a bacteriocin, which is a protein that prevents other cells from growing. You can think of a bacteriocin as being a very narrow-acting antibiotic, like the bacteriocin produced by E. coli only inhibits the growth of Salmonella and Shigella. It does not prevent the growth of any gram-positive cells, nor does it prevent the growth of some gram-negatives, like Enterococcus faecalis, a microbe which is part of a normal microbiota, probably the number one normal microbiota in the gastrointestinal tract. Okay, so the bacteriocin of E. coli only inhibits the growth of certain microbes, such as Salmonella and Shigella. Other microbes are not inhibited. I've always wondered why they do not use bacteriocins as an antibiotic. And recently, a few years ago, I read a paper that they were using bacteriocins as a experimental antibiotic, meaning when someone got salmonella infection, they were given a bacteriocin instead of an antibiotic. And this will be beneficial because instead of getting an antibiotic which harms all gram negatives to get rid of the salmonella in your intestines, you use the bactericin, and it would only get rid of salmonella and shigella. It would not get rid of any other gram negatives in your intestines. And so that would be a very good thing to use. Any question about any of that? All right. Probiotics is the consumption of live microbes intended to exert a beneficial effect. It used to be, I'm not sure if they still do this, but when I was given oral antibiotics, the doctor would tell me, and you should probably eat some yogurt with live cultures to help establish the normal microbiota in your gut back again. Another advantage of eating yogurt with live cultures is it reduces the chance of getting a yeast infection. And that is the bacteria that you consume in the yogurt colonizes you and then occupies the niche, competes for resources, and then the yeast cannot get established and then cause a yeast infection. Any question about any of that? All right, the relationship between the normal microbiota and the host is one of symbiosis. Symbiosis is where two different species of organisms or two different populations live together. It can be in harmony, but it doesn't have to be. It can be pathogenic in symbiosis. The normal microbiota and the host live in symbiosis with each other. There are three forms of symbiosis. The normal microbiota either has mutualistic symbiosis or commensalistic symbiosis. Commensalism is where one organism benefits and the other organism is unaffected. An example of commensalism is each of us has one nematode living in our eye. Or I should say, I guess that's the average, not necessarily each of us. The nematode does not harm your eye, does not eat eye cells, does not harm the individual. I'm assuming the nematode is eating the bacteria that fall into your eye. The nematode gets a benefit, and that is it gets a home from the host, meaning it makes its home in the eye. But the nematode does not do any harm to the host. So it's commensalistic relationship. Any question about any of that? Some of our normal microbiota form a commensalistic relationship. The other normal microbiota form a mutualistic symbiosis, and that is where both organisms benefit. In our gut, we have E. coli. E. coli benefit because they get a home and then we feed them, meaning food in our gut, E. coli can digest. We benefit because E. coli helps us digest our food. So we get a benefit out of that as well. And that's a mutualistic relationship. Any question about any of that? Now, a human pathogen forms a parasitic symbiotic relationship. Like when Staphylococcus aureus gets on our skin and then causes a skin lesion, it is a parasitic symbiotic relationship. Staphylococcus aureus benefits, it gets a home, it finds a home living on us, and it also damages us, meaning it feeds off of our cells. So that benefits the Staphylococcus aureus, and that does harm to us. And that would be actually all human pathogens are a parasitic symbiotic relationship. Any question about any of that? All right. The normal microbiota does not normally cause a human infection. So it does not normally cause disease. However, under some circumstances, the normal microbiota may cause disease. Some normal microbiota can become what is called an opportunistic pathogen, where they normally do not cause disease, but they may cause disease under certain circumstances. Like when the individual is not healthy, the normal microbiota can then cause disease. Like in an AIDS patient, part of the normal microbiota can grow, and then they have no immune system to keep the normal microbiota in check. And then the normal microbiota can become opportunistic, causing disease. A cancer patient taking a chemotherapy will have a weakened immune system, and the normal microbiota can grow. And the immune system doesn't keep the normal microbiota in check, so it can cause an opportunistic infection. Whenever the normal microbiota grows in places where it should not, it will be an opportunistic microorganism, like E. coli, normally found in the gut. But if it starts growing in the ear, it can cause an ear infection. Staphylococcus aureus is normally, well, in 50% of Americans is found in the nose and the throat. And in the nose and the throat, it tends not to cause an infection. But if it grows on the skin, it can cause an infection. And if either of these grow in the lungs, it would cause pneumonia. And in which case it would be an opportunistic pathogen. It is part of the normal microbiota, at least E. coli. But when it's growing where it should not be, it will be an opportunistic pathogen. Any question about any of that? All right, if not, let's talk about Koch's postulate. We're going to talk about Koch's postulates 3 times this term. This will be at least the second time. I remember we talked about it in chapter 1, briefly talked about Koch's postulate. Koch's postulates were the step Dr. Koch used to show that the bacteria Bacillus anthracis caused the disease anthrax. And he decided to publish it because the steps that he used, let me see if I can blow that up, that's better, are used to show that a given microbe causes a given disease. The steps of Koch's postulate is, first, you obtain a pathogen from a sick individual, or in this case, a sick bunny. You have to grow it up in tissue culture, and you have to isolate it and grow it as a pure species. So you isolate it as a single colony and then streak it out on another plate, isolating it from a single colony, then grow it up as a pure culture, take the pure culture, and then inject it into another individual or another bunny in this case. And if this bunny becomes sick, while the bunny is turned on its side, the bunny is going to become sick. Ah, the poor bunny. And assuming that the disease symptoms are the same as the first, that's very good on showing that this microbe causes this disease. But Dr. Cox said you got to take it one step further, and that is from this sick bunny, you have to isolate the microbe that you injected into the bunny. And if you do that, you've fulfilled Koch's postulates, and you can say that this microbe causes this disease. Any question about any of that? So Cox postulate is what we prefer to use when we want to show that a given organism causes a given disease. But there are exceptions to Cox postulates. Like, what do you do with microbes which have unique culture requirements and cannot be grown in artificial media, cannot be grown in tissue culture? For, excuse me, for over 100 years, the bacteria which cause syphilis, they were not able to grow it in tissue culture. They could never figure out what they needed to add to the culture to get the syphilis to grow in tissue culture. So for 100 years, they figured out which microbe caused syphilis, but they could not use Koch's postulate to prove it. Until recently, I read within the last, I think it's 5 years, they figured out what they need to add to tissue culture to get syphilis to now grow in tissue culture. Another one which they may not have figured out what they need to add to get it to grow is Bartonella henselae, the microbe which causes cat scratch fever. And it's possible within the last 5 years, they've been able to get it to grow in tissue culture. I don't know, but it's likely, at least for the last, up until the last 5 years, they never figured out how to get it to grow. And it's possible it still cannot grow in tissue culture. But what do you do if multiple organisms can cause the same disease, signs, and symptoms? If you remember, Koch's postulate states all organisms that have this disease are caused by this one microbe. An example is the human cold. Not only is it caused by different species of viruses, but there's actually 3 different families of viruses that cause the human cold. So you cannot use Koch's postulate to prove that a given viral species causes the human cold. And then some species can cause different diseases or different disease conditions, like salmonella can cause salmonellosis, but it can also cause, I think it's typhoid fever. Is that correct? Does anyone remember? Yeah, it's typhoid Mary. So it's typhoid fever. I'm sure of that now. I just remembered typhoid Mary is the nickname for the person who was spreading around typhoid fever. And so we'll stop that and share my screen again. Where are we? So what do you do if the species causes 2 disease signs and symptoms? You can't use Koch's postulate because Koch's postulate states all diseases are caused by this one microbe. In reality, like I said, this one microbe causes 2 different diseases. And then what do you do if moral and ethical conditions keep you from injecting the disease-causing organism into a human for a human-only disease, for example? Like, for example, HIV has never been injected into humans to test whether HIV causes AIDS. And yet we know that HIV causes AIDS. Anyone have a guess as to why we know HIV causes AIDS? Anyone? What they did was they studied very large epidemiological studies, and then they followed this group over time. And some of the individuals who are HIV negative on their own became HIV positive. You then follow those HIV positives over time, and some of them actually, back then when they first discovered this, almost all of them eventually developed AIDS. Okay. And so you can use very large epidemiological studies to show that a given microbe causes a given disease. But the preferred way is to use Koch's postulates. Okay? Any question about any of that? If not, let's talk about classifying infectious diseases. There are 3 terms you need to know. A symptom is a change in the body function that is experienced by a person as a result of disease. But nobody else can measure or know about that symptom, meaning it's subjective. It's not apparent to an observer. Like if a patient has a headache, there's no way for anyone to confirm that that patient has a headache because you can't measure it. And that's a symptom. A sign is a change in the body that can be measured or observed as a result of disease. So a sign is objective. Like if a patient has a temperature of 102, anybody can verify that the patient has a temperature of 102. You just put a thermometer in them and you get the temperature. Any question about the difference between a symptom and a sign? Now you'll learn that in the clinic, the terms symptom and sign, as well as in general usage, are used interchangeably. But a symptom really is only a change that is subjective and cannot be verified by anybody. A sign is a sign of illness which can be observed and measured, such as the temperature. A syndrome is a specific group or signs of symptoms that characterize a disease. For example, the signs and symptoms characteristic of AIDS is called the AIDS syndrome. Any question about any of that? A communicable disease is any disease that can be spread from one host to another. Example, measles, tuberculosis, and chickenpox are all communicable diseases. A contagious disease is a disease that is easily spread from one host to another. Can anyone give me an example of a contagious disease? A disease which easily spreads.
Chickenpox.
Chickenpox is a contagious disease. Anyone else? There's actually lots of them.
COVID-19.
Yeah, COVID-19, a contagious disease. It's more difficult to come up with a communicable disease which is not contagious, meaning a disease which spreads, but it does not spread easily. Does anybody know of a disease which is communicable, but it is not contagious? Anybody have a guess?
Would it be like an ear infection or a UTI?
Say again, I didn't hear the first part.
Like an ear infection or a UTI?
It's possible. It depends on what is causing that disease. Now,
Okay.
obviously, most UTIs are not going to spread patient to patient. Okay. I mean, it's possible, but they're not likely to spread patient to patient. But an ear infection could spread patient to patient if it's like a viral infection causing the ear infection, it can spread among children. But if it's a bacteria that is causing the ear infection, it's not likely to spread from child to child, just because the bacteria do not normally come out of the ear.
So would it be like tuberculosis?
Ah, there's a good one. Tuberculosis is actually a communicable disease,
Okay.
but is not contagious. I was going to say there was one actually listed here that is a communicable disease, but it is not contagious because it can spread, but it doesn't spread easily. You need long-term exposure to pick up TB. Another classical case, in fact, probably the best case, is leprosy. Leprosy does not easily spread from one patient to another. You need long-term exposure, and only something like 5% of the population can even pick up leprosy. Something like 90% of the population cannot pick up leprosy from another patient. I'm not sure if that's leprosy in general or they can't pick it up from another patient, but the point is that leprosy is communicable, but it is not contagious. It does not tend to spread. Okay, any question about the difference between communicable disease and contagious disease? There is a non-communicable disease, and this is a disease that does not normally spread from one host to another. Tetanus and botulism are two non-communicable diseases that do not spread from one host to another, at least in normal circumstances. I can think of one case where tetanus could spread among the population, and that is if somebody were to get tetanus and then a cannibal were to eat this patient and not cook the patient, that cannibal likely would get tetanus. Okay, oops, didn't mean to do that. But normally tetanus does not spread from patient to patient. So normally tetanus is a non-communicable disease. Any question about any of that? Okay, to understand the full scope of a disease, we need some details about its occurrence. When we're talking about occurrence, you can separate diseases into two groups, the incidence and the prevalence. And you can call the incidence the new cases of disease and the prevalence both the new cases and the old cases. So the incidence is the fraction of a population that contracts a disease during a specific time. Like when we have the flu season and people talk about the number of people currently having been exposed to the flu or gotten the flu, we're usually talking about the incidence. We do not usually talk about prevalence of the flu, because if we were to talk about the prevalence of the flu, probably 100% of the population would be getting the flu. Very few people would not ever get the flu. But in some diseases like AIDS, we tend to talk about the disease with the prevalence, meaning when we're talking about AIDS or HIV, we're talking about the fractions of the population having the specific disease at a given time, including both the new cases and the old cases. So in 2007, the incidence of new cases of AIDS were there were 56,300 in the United States, whereas the prevalence in 2007 was 1,185,000 cases of AIDS. Okay. I think with COVID-19, they usually use the prevalence, meaning the population of the United States, which has gotten COVID-19, which now is probably getting close to 100%, but it last I checked, it was less than 100%, something like 85% of the population have had COVID-19. So with some diseases, we may switch to start talking about it, the incidence. Like I said, with flu, very common diseases, we usually just only talk about the incidence. All right, any question about any of this? Few more terms. A sporadic disease is a disease that occurs only occasionally in a population. For example, typhoid fever in the United States is usually said to be a sporadic disease, that it only happens occasionally. Another example would be malaria in the United States. It's a sporadic disease. It only happens when somebody goes elsewhere in the world, picks up malaria, and then brings it back to the United States. And that's not entirely true. I think it was last summer, malaria was actually spreading in 3 states in the United States. What happened was somebody brought malaria to the United States, and then mosquitoes in the Gulf states can spread malaria, and the mosquitoes bit that individual and then bit somebody else. And so mosquitoes in Texas and Florida and one other southern state, I think it was North Carolina, but I'm not certain of that state. But Texas and Florida, mosquitoes were actually spreading malaria in the United States last summer. That does not normally happen. Mosquitoes do not normally spread malaria in the United States, but they can. At least in the Gulf states where the mosquito is present that can spread malaria. Any questions about any of that? An endemic disease is a disease that is constantly present in a population, such as the common cold is endemic to the human population wherever you are on the planet. Malaria is endemic to the tropics. So if you go to the tropics, you should take steps to prevent getting malaria, because malaria is found in the tropics and it's always spreading in the tropics. An epidemic is a disease acquired by many hosts in a given period of time in a certain location. For example, occasionally we have influenza, significantly higher numbers than expected, in which case it's an epidemic of influenza. When COVID-19 first hit China, it was an epidemic in China. An epidemic may spread worldwide, in which case we don't call it an epidemic anymore. We call it a pandemic. Usually the flu starts in one country as an epidemic and then spreads worldwide. Certainly COVID-19 did. It started in China and then spread worldwide and became pandemic. So the only difference is pandemic is 2 or more continents. Usually it becomes worldwide. An epidemic is it's only on one continent. Any question about any of that? I can mention a case of an epidemic that never went to a pandemic, and that is SARS-2, meaning the second time people got SARS. It was only an epidemic. It only stayed in China, and maybe it spread to like Korea or someplace else in Asia, but it was only in Asia. And then they got it under control, and fortunately SARS went extinct, at least in the human population. The first time SARS came in 2003, it did go pandemic. It did spread to other countries in the world. I think the United States and Canada were 2 countries that did enter. But fortunately, the health community got control of it, isolated those sick individuals, and nobody else got SARS. And SARS went extinct in 2003. But it did hit the world population once again, starting in China once again. And we'll talk a little bit about that when we're talking about COVID-19. Any question about any of that? All right, when we're talking about disease, it's important to talk about the severity or the duration of a disease. It's a useful way of defining the scope of a disease to talk about its terms of severity or duration. Excuse me. We can split diseases into two categories, an acute disease and a chronic disease. An acute disease is a disease where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. A classic case of an acute disease is the human cold. The disease develops rapidly, the patient becomes sick very rapidly, shedding very large numbers of virus very rapidly. And the patient then soon gets better. The disease is short-lived and the disease goes away, usually within a week. A chronic disease is a disease that develops slowly and persists for a long period of time. A case of a chronic disease would be mononucleosis, where the patient initially gets an infection. They usually have signs or symptoms of a cold. The disease develops slowly, gets worse over a month or a few months. The disease persists for a long period of time, usually several months. The patient usually gets worse, and then they develop mononucleosis. In the case of mononucleosis, the patient usually does get better. That's not always seen in a chronic disease. Like hepatitis B can be a chronic disease. The patient initially gets a hepatitis B infection. They might have hepatitis, and then it becomes chronic. And they usually do get better, but then the hepatitis comes back, and then they get better and the hepatitis comes back. The point is the disease develops slowly, persists for a long period of time. I think hepatitis B, some patients never throw it off. And usually each infection or each hepatitis bout, the patient gets worse. A chronic disease. Another chronic disease is tennis elbow. Some tennis players wear out their elbow, usually with old age. And each time they play tennis, or the tennis elbow just gets more and more damaged and then gets worse with time. A case of a chronic disease. That never gets better. Tennis elbow never gets better. Any question about any of that? All right, you can have something in between an acute disease and a chronic disease. You don't need to know about it. It's called a subacute disease. The symptoms are between an acute and a chronic disease. An example is subacute sclerosing panencephalitis. And I'm sure most of you have never heard of subacute sclerosing panencephalitis. So like I said, you don't even need to know about subacute diseases. When we're talking about disease, it's important to talk about the extent of host involvement in the disease. Infections can be classified according to the extent to which the host body is affected. You can have a local infection, and that is the pathogen is limited to a small region of the body. For example, a skin boil, that's the only region where the pathogen is, on the skin boil, causing the skin boil. It is not elsewhere in the body. Or an abscess of the skin, a local infection. This differs from a systemic or generalized infection, where the infection can spread throughout the body. When a patient gets the measles virus will become a systemic infection and spread throughout the body. Any question about any of that? A focal infection is a systemic infection that began as a local infection. For example, MRSA may initially cause a skin boil, but then the skin boil gets worse and worse, and the MRSA can get into the blood. And then the blood can spread the MRSA from the skin throughout the body. And then the local infection can become systemic, and we call that a focal infection. Any question about any of that? Bacteremia is presence of the bacteria in the blood. This differs from septicemia, and that's where bacteria are actually growing in the blood, where bacteria are actually growing in the blood. Usually when a clinician runs a test, they will only be able to see the bacteria in the blood, so it's bacteremia. But specific tests can isolate septicemia, where the bacteria are actually growing in the blood. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Any question about any of that? All right, a little bit more about the extent of host involvement. The state of the host resistance also determines the extent of the infection. A primary infection is an acute infection that causes the initial illness. Like when the patient first comes down with the cold, they usually get a sore throat as their primary infection. A secondary infection is an opportunistic infection after a primary infection. So the primary infection is the predisposing infection, which has weakened the host, allowing the secondary infection to get established. And the secondary infection is often opportunistic, meaning it can be the normal microbiota that does not normally cause disease, but because the host is weakened by the primary infection, it can then cause a secondary infection. One secondary infection is a child comes down with a cold, which is the primary infection, and then the child gets an earache where bacteria then start growing in the child's ear. The bacteria is the secondary infection, which happened in the child because the primary infection weakened the child, and then the bacteria got established in the ear. Any question about any of that? That can happen to an adult too, but it's more likely to be seen in a child because of the tubes of the child ear. What do you call those? The Eustachian tubes or something like that. They're more flat. They don't go down. In an adult, they tend to go down. And so in an adult, they tend to drain better than a child. And that's why children, once they have a cold, they're at a greater risk for getting an ear infection. A subclinical disease is when a patient gets the disease-causing organism. However, the patient has no noticeable signs or symptoms of disease. This is an inapparent infection. And we've talked about it before. We call it an asymptomatic disease. Here it's called a subclinical disease. Some people can carry polio, the polio virus, but they never get signs or symptoms of polio. And they can spread the polio virus from one person to another. So they are a carrier, but they themselves never have any signs or symptoms of the disease polio. And here's one for you because lots of you are young. Most of you are young. Actually, all of you possibly. I haven't looked at the age of people, but I know most of you are young. Young people and children, about 50% of you can get COVID-19. And the virus will be subclinical or asymptomatic. So you have no signs or symptoms that you have COVID-19. But you can spread the virus to other people, like your grandparents. And when they get it, it could be a very serious disease. Any question about subclinical diseases? If not, I'm going to stop here. I'll be in the lab for 15 minutes to answer questions. But if you don't have any questions, you don't need to show up. Any questions before I log off? All right. I will see you next time.
You said you're gonna log back in what time?
I'll log in at the normal time at 6:30 in the lab.
Okay, sounds